So these three we mainly need to remember, okay? The parietal cells will secrete hydrochloric acid, okay? It activates pepsin, okay? So chief cells will secrete, so pepsin is an inactive form, pepsinogen is an inactive form of pepsin, and then so hydrochloric acid will activate it, okay? Mainly the hydrochloric acid, remember the stomach, mainly is not for digesting anything, it's not to absorb anything, but to denature the protein, okay? So the acid will denature the protein, destroy the bacteria. It will secrete intrinsic factor, okay? Remember we saw that earlier. So we can't really, so if we eat some steak, protein from a cow, we really can't use the proteins from the cow. We have to denature it, break it down into amino acids, and regroup it into something that we can use, okay? Chief cells, again, it will secrete pepsinogen, which is an inactive form of pepsin, and then the parietal cell will activate the pepsin. The endocrine will produce gastrin, okay, and other hormones. Gastric secretion. Brain, stomach, and small intestine. Neuro, higher brain, local reflexes, and autoimmune system, autonervous system. Parasympathetic stimulation of digestion, and sympathetic will inhibit digestion. Hormonal, we will see gastrin stimulate. GIP, CCP, and secretin will inhibit. So here's the cephalic part, which is the brain, the gastric part, okay? So stomach is distended, it got bigger, okay? And all these things will happen, and it will stimulate the stomach secretion, okay? Too much acidity, emotional distress, okay? Sympathetic nervous system will activate. Remember, sympathetic nervous system will stop digestion, okay? Inhibit stomach, okay? Here's the intestinal part. Presence of low pH, intestinal gastrin released in the stomach, stimulate the stomach secretory activity, and here this part inhibits distension of the duodenum in the presence of fatty acid, in the presence of chyme. So when you have food in there, okay, local reflex, phyloid sphincter, enterogastric reflex, it will inhibit stomach secretory activity, having slow it down. Okay, let's digest it first. Oh, it's all here. So before food enter the stomach, he's having some stimuli, aroma, taste, the sight of food, thought of food. Response, stimulate of the gastric gland via parasympathetic to release mucus, hydrochloric acid, pepsinogen, IF, and gastrin. Oh, by the way, IF is intrinsic factor, P12, remember? Okay, now at the gastric phase, food enter the stomach. It goes 3 to 4 hours. Okay, partially digested protein, rising of the pH, response, the parasympathetic increases gastrin, which target the parietal cells, which produces HCl, and the smooth muscle stimulate. Intrinsic factor, pH. If the pH drops too low, hydrochloric secretion will stop, and the mucus increases. So remember, this is when the pH is too low, when the pH drop too low, which means it's becoming way too acidic. Okay, then the body will stop secreting hydrochloric acid to not digest the stomach, and the mucus will increase to protect the stomach. And here, a stimuli, the chyme will enter the intestines. In the presence of acid, okay, fat, partially digested protein in the duodenum. What's going to happen? Remember, whenever there is acidic, okay, CCK will come in. It will stimulate the enterogastric reflex, sympathetic to tighten the pyloric sphincter. Intestine will release secretin, CCK, GIP to inhibit gastric secretory activity and movement to protect the small intestine from acidity. You remember in the pancreas, it will secrete some bicarbonate so that it doesn't digest its own, okay, to neutralize the pH. Digestive function generally takes 2 to 4 hours. Fat takes longer to leave because digestion in the small intestine is slow. Satiety is when you're satisfied with the food you ate. Mechanical digestion, we saw that already. Chemical digestion, we saw all this already. Okay, start with salivary amylase. Okay, that's the first part of the digestive system. So this is, we're going to start with the small intestine. Small intestines, where most chemical digestion and nearly all absorption occur. Okay, in the small intestines. The large intestines mostly absorb water. Okay, so the small intestine is made of 3 regions. Okay, the first one is duodenum. Okay, we saw duodenum already, where the chyme goes in. Okay, and here's the stomach. Okay, first section is the duodenum. Second section is the jejunum. Okay, jejunum. And the third section is the ileum. Okay, ileum. It's got more vessel and lymph node join the cecum. Cecum is the beginning of the large intestine. Okay. Mesenteries have just been promoted to an organ. They're a complex double layer serous membrane right on top of the intestines. And remember, we saw, I think it was last chapter, we saw there is a MALT, which is mucosal associated lymph tissue, and Peyer's patches, which are little nodules all over here. They keep bacteria from entering the bloodstream. Okay. Histology, if we look at it, even smaller, okay. Absorption, this is adapted for absorption, so this is a section of the small intestine. Plica circulae, this is a circular fold of the mucosa and submucosa. So when the chyme comes in here, it doesn't go straight through. Okay, it circulate, it kind of churn and fold because of these circular fold, okay, pattern. This is called plica circulae. So that it doesn't go straight through, and it touches everything, so that it increases absorption. Okay. So here is a magnifying of a villi. Okay. Villi is for increased absorption. And on the villi, there are villi, okay, called microvilli, even for even more absorption. Okay, so each villi has a little hair-like thing. So these villi are already small, and these microvilli are even smaller for absorption. So each villi has a lacteal. You guys remember the lacteal? It's for fat absorption. And each villi has an artery and vein for carbohydrate, transport of carbohydrate. Okay. Oh, yes, microvilli. Brush. Oh yeah, Peyer patches on top. Here again is the villi, and there's microvilli, lacteal, and the arteries. Okay. The villi projection projects into the lumen. Okay. Again, here's the villi, and the lumen is where the food will go through. It kind of brushes. It brushes it. Okay. Oops. Okay. Sorry about that. So the food brushes right through there, and then it will absorb some of the things. Again, in the submucosa, the duodenal gland secretes a bicarbonate-rich alkaline mucus. Why? Again, is to protect, so that it doesn't digest the small intestines itself from the acid. So in the intestinal gland, there are different cells. Okay, the S cells, it will secrete secretin. The I cell will secrete CCK. Okay. Pannus cells or granular cells will secrete lysosome and for phagocytosis. Again, there's bacteria and other things in here. Okay. But you'll see also M cells will secrete motilin. So M for motilin. Motilin again is to mobilize. Okay. And K cell will help secrete the pancreas, insulin. So here is peristalsis. Segmentation, localized mixing of the chyme and secretion, as well as pushing the fluid against the mucosa for absorption. When most absorption has occurred, peristalsis, migratory motility complex due to motilin, propulsion. So when most absorption has occurred, so here is it actually doesn't just go straight down. Okay, it goes up and down, up and down, up and down like that. Okay, movement usually takes 3 to 5 hours. Okay, we saw motilin. Motilin, what cell is it coming from? M cells. Okay, M cells. Relaxes control this and prevent blockage. Stomach will signal other part of the tract when food enter and gastrin is released. The gastroenteric reflex increases peristalsis in the small intestines. So once food comes in, it's going to tell the small intestine, hey, move away. Okay, time for more food to come. Gastroileal reflex, the reflexes of the ileosphincter, which is usually constriction and increased peristalsis in the ileum. So the ileocecal sphincter, it is